Much. I am really honored to be asked to talk with you today. You should know that everybody in the United States is very concerned about the situation in Ukraine, and we follow it all the time. But we're not the ones living through it as you are, and our thoughts are constantly with you. And although maybe this talk will be a little bit trivial in comparison to what you're thinking about all the time, I hope it could be maybe a little bit enjoyable for a while. So let me start. And I've given my talk a rather unusual title, The Continuing Need for Useless Knowledge. And this title actually comes from a educator in the United States named Abraham Flexner, who wrote an article in 1939 in which he said that we needed the we needed useless knowledge. I sometimes wonder whether our conception of what is useful may not have become too narrow to be adequate to the roaming and capricious possibilities of the human spirit. He wrote this in this article in 1939 in Harper's Magazine. And in it, he says, from a practical view, intellectual life is on the surface a useless form of activity. In this paper, I shall concern myself with the question of the extent to which the pursuit of these useless satisfactions proved unexpectedly the source from which undreamed of utility is derived. And he starts off the article with a very interesting story. He says that he is talking to the industrialist George Eastman, and he asked George Eastman, who do you think is the most important scientist in the world? And Eastman, without any hesitation, says that that is Marconi, the inventor of radio and wireless. And Flexner writes that he says to Eastman, whatever pleasure we derive from the radio or however wireless and the radio may have added to human life, Marconi's share was practically negligible. He was inevitable. The real credit for everything that has been done in the field of wireless belongs to Professors Clerk Maxwell and Heinrich Heinz, because these are the people that developed the understanding that led to the application of radio and wireless. And we see this repeatedly in many cases. I will give you a couple of examples of these. One of them, clearly we've seen, you probably are carrying around in your pocket of one for another, a smartphone. And the Quartzoft company several years ago made this wonderful infographic, which I'm sure you cannot see here, but the link is here. And if you go to that site, you can see this chart in all of its glory. But if we just look at a couple of areas in this chart, you'll see what this is all about. Because what this is how do we get a smartphone? How do we get the iPhone with its Wi-Fi and battery and touch screen and computing and internet connections, its display and its camera? What were all the scientific discoveries that led to this? And as you go down the chart, you find that there's hundreds of different discoveries. And you go further and further down the chart, and you get finally to the discoveries here at the bottom of the first binary system, or working on the first lens, or working on the geometry of vision, or looking at or using zero in a calculation, or being able to produce copper for the wires. All of these things are the basis of something that looks like it's invented out of nothing. So all of these things are built on lots and lots of what at the time is probably referred to as useless knowledge. So even more recently, the vaccines against COVID-19. In this editorial, Anthony Fauci wrote, "What is not fully appreciated is that the starting point of the timeline for the SARS-CoV-2 vaccines was not the 10th of January 2020, when the Chinese published the genetic sequence of the virus. Rather, it began decades earlier out of the spotlight." And that's in fact where most of the useless information, the useless knowledge is derived, without people making a big to-do about it. And I want to show you one of my favorite videos about this importance of basic research and what it can mean. So let me just start this up. This is going to take about 4 minutes.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the National Institutes of Health, NIH, are 2 government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technologies, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come? Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research. We are the scientists who depend on you.
And we are excited for the future that lies ahead.
Wonderful. So if it's important to make discoveries, how do we make discoveries? And obviously we do experiments, but I think that's not enough. The Nobel Prize-winning physicist Enrico Fermi is quoted as saying this very interesting line. He says, if the result confirms the hypothesis, then you've made a measurement. If the result is contrary to the hypothesis, then you've made a discovery. So while it's good to have hypotheses, one has to be alert to all the other possibilities and really look at what happens when things go wrong, which I think is a nice way of thinking about how we do science. And this has been acknowledged in many ways because people that have made accidental discoveries, but then were able to follow through on these discoveries, have really changed our world. There have been several Nobel Prizes based on this. For example, we have the Nobel Prize to Robert Wilson and Arno Penzias for the discovery of the background radiation of the cosmos that they did by not really looking for it, but realizing that they couldn't get rid of the background in their microwave telescope that they were using. Or the discovery of place cells by John O'Keefe, who in his Nobel Prize lecture says that he was doing an experiment and by accident, he put the electrode in the wrong part of the brain and suddenly discovered a very interesting type of cell that was responding and then followed up on it. The third example I want to give is one that I'm more personally involved in, not that I did the initial work, but that it changed the way my life has gone. And that's the discovery of the green fluorescent protein by Osamu Shimomura. Shimomura, who died a couple of years ago, was a remarkable biochemist and has had a remarkable life. And if you have a chance to read his memoir, Luminous Pursuit, it's well worth reading. At the age of 16, he is in high school. He's told he has to quit high school and go and work in a factory. The factory is outside the town he was living in, which saved him because the town was Nagasaki, Japan, and he was working in that factory when the atomic bomb destroyed the city. He went home, he helped people, he couldn't go to school for several years until they rebuilt and made the first university near him, then went to work as a technician. Made an interesting discovery as a technician, which I don't have time to go into, but that led to his lifelong interest in how different organisms can generate light. Now, many different organisms generate light, and they do so differently from one another, whether they're glowworms or fireflies, or in this case, jellyfish or crustaceans. They all biochemically do it differently. And he started his studies working on this jellyfish, Aquaria victoria. And if this were the standard science story, it would be of a person who knew exactly what they were doing, had a hypothesis, and developed it. And although Osamu was a spectacular biochemist, I have to say that the entire year in 1962, when he was studying this, that entire summer, he ground up lots of jellyfish trying to find the molecule that was generating its wonderful green light. And all the experiments failed. And they failed time after time after time. Towards the end of the summer, his experiments has failed again. And he takes the prep, because it's late at night, he wants to go home and have dinner, and he throws the prep into the sink. Sink had some seawater, other things probably as well. He turns off the light and he's just about to leave when he looks back at the sink. And to his great surprise, the sink is glowing brightly, something he had never seen in the preparation before. He's a little surprised at this. He goes back and he thinks about it, and he realizes that the seawater in the sink had something he had never tried in his experiment, and that was calcium ions. So he goes back after a couple of days, redoes the experiment, but this time he adds a solution of calcium ions to the preparation. And when he does it, the lights come on. So now he has a way of purifying the protein. He names the protein after the jellyfish. And this light producing protein is called a quorin. And so a quorin plus calcium gives light. Well, that's all very nice and a very accidental discovery. I like to say that this is the real scientific method. People throw things on the sink or on the floor or on the lab bench, and then all of a sudden the experiment works. But in this case, although the experiment worked and he found the protein that produced light, there was a problem, an unexpected problem. The light was the wrong color. The light was blue, whereas the jellyfish produced a green light. And although he added calcium to all the other samples, no other sample gave green light. So he thought about this for a while and he realized maybe there was something that converted the blue light to green. And so he took a handheld lamp and shone a blue light on all of the samples. And when he did that, he found that there was a completely other protein that when blue light hit it, it gave out light that was green. This is called fluorescence. And so he called the protein, the green protein, in a footnote. In fact, it's the only footnote I know of that has won its author a Nobel Prize. He called it the green protein. We call it the green fluorescent protein, or GFP. And GFP, when a quorin is activated by calcium, the light that would go to make blue light is now converted and you get the green light of the jellyfish. Now, when I was 1989, I was sitting in a seminar. I was listening to a seminar and someone told me this story. I wasn't working on anything like this, but I was interested in how genes were activated in different nerve cells. And there were a number of ways of looking how genes were turned on, but I realized listening to the story that this would be a wonderful tool to show where genes were turned on. Because I realized that all you needed, you didn't need a quorin, you didn't need calcium, you just needed blue light, and you would be able to see wherever that protein was by looking at green light. In other words, this would provide a lantern that would allow us to look at the cells. So I was very excited about this and eventually worked with a graduate student, Gia Eskerkin, and a collaborator who was isolating the DNA for GFP, Douglas Prasher. And we put the gene for this protein into bacteria, irradiated them with blue light, and were able to see where the bacteria were because they fluoresced green. We then later put it into the animal that we work on, a small worm called Cenorhabditis elegans, work on a set of 6 cells. We had a gene that was expressed only in those 6 cells, and we could see them in the animal, where before it would have been very difficult to see the cells. And more than that, we could see them in living cells. We gave this out to everybody that wanted it. And people started putting it in other tissues. So people put it in fruit flies, or they put it in zebrafish, or they put it in mice, all sorts of animals and plants and bacteria and so on. And one could see cells outlined because the GFP went everywhere in the cells. Now, I want to show you one of the really wonderful aspects about this. Because we're only shining blue light onto a specimen, we can do something that was very difficult before GFP. We can actually watch things happen. Life is dynamic, but before GFP, one had to fix the preparation, which meant it was killed and permeabilized. You got just a snapshot in time. But now people were able to look at how things changed over time. So I want to show you two short videos by Rosalind Silverman Gavrella, who at the time was a graduate student in Canada, and she was working on fruit fly development. And on the movie on the left, she has put GFP into a protein that is part of the spindle, the part that separates the chromosomes when a nucleus or a cell divides. And so if we look at this movie, it's very sped up, but it's going to go through 4 rounds of division. And you can see the spindles forming now, and then they break apart. And this goes through 4 things. This is very much sped up. But I hope that you see something really quite remarkable here, and that is all the divisions are happening at the same time. We're able to see that. And that immediately raises questions as to how that actually takes place. Now, the movie on the left, although it has some color, is really falsely colored. And it's falsely colored because the more GFP that's present, the more towards the red end of the rainbow is the designation. So blue is the least, green a little bit more, yellow a little bit more protein, orange even more, red the most. And here we'll watch the same divisions, but now we're looking at GFP, not part of the apparatus that separates the chromosomes, but rather it is attached to a small signaling peptide, and this signal brings the GFP into the nucleus. Well, during division, there is no nucleus, so the GFP will be everywhere. So as we look at that picture, here you can see the nuclei break down. Now the GFP is everywhere in the embryo. The nuclei reform and it gets sucked up into the cells. And then we go through another round of division where the nuclei break down. Now it's all over the cells and so forth. Now, this is not synchronous. It seems to go from the lower right-hand corner to the upper left-hand corner. And we think that that may be because this was a slightly damaged preparation. But at least we have a very fine way of looking at what is happening in the cells. Now, GFP, like virtually any discovery, leads to lots of other discoveries. Some of them are more understanding of fundamental research, more basic research, more useless information. And one of my favorite aspects was the discovery by Cliff Bregman and Tony Hyman in 2009 of a new type of component in the cytoplasm of cells, as if there was oil and water within the cells. These are called phase-separated particles. And it has become one of the most exciting new areas in cell biology. But there's also applied research. And I want to give you an example of thinking about how viruses go from one cell to another. When in introductory biology, we usually teach that cells are infected by a virus, then the cell explodes. And in that explosion or lysis, the virus goes everywhere and infects other cells. But does that really happen all the time? And the answer is no, it doesn't. And here's an example of the AIDS virus, HIV. It's infecting a cell, and in the virus, we've put the researchers put the coding sequence for GFP. So this is an infected cell, and you see that there was a small particle that went from one cell into an uninfected cell to now infect that cell. No explosion. What are the implications of this? Well, if there's an explosion, maybe antibodies can bind and get rid of the virus. But if it's going from inside one cell to inside another, then it's sort of protected. And it immediately suggests that there should be different ways of looking at intervening in the spread of the virus from one cell to another. Now, in reality, although we sometimes talk about fundamental research giving rise to applied research, it really is a two-way street. We go back and forth. But in addition to this, there are some rather unexpected things that come up. And I want to give you some ideas about some of the things that have been unexpected for GFP. So several years ago, I went to Japan and met some researchers there who had decided to put GFP into silk moths. And they generated silk that was fluorescent because it had the GFP and other fluorescent proteins as well. And I'm not sure exactly the usefulness of something like this, because I think you have to carry an ultraviolet lamp with you in order for people to see the wonderful fluorescence. Another example is that people have turned cells into lasers by using GFP. Bob Berlage, several years ago, designed a bacterium that would fluoresce. It would make GFP and fluoresce, but only in the presence of the explosive TNT. Why did he do something like this? Well, he was able to have a friend bury, he knew that landmines leak TNT. And so he was trying to find a way of detecting landmines so that people were not injured or hurt. And still is something that lots of groups are working on. He was able to find it by having his friend bury landmines disconnected in a field. He then sprayed it with bacteria, and he was able to see where they were by the fluorescence in the evening. This is still something that's, as I say, is being worked on because people really want to make sure there are no false negatives. Another example, GFP has been used in the arts. The Brazilian artist, Eduardo Koch, had a French biotech company generate a rabbit, a family pet, that had GFP in it. And he would bring this to his art shows and get people to talk about the connection between art and science. And the final example I want to give here is one in which GFP has made it into the movies as an explanation for why this particular character has his green color. Let's get in here. So let me go on from here. Whoops, sorry, went the wrong direction. The third person to share the Nobel Prize with us was Roger Chen, who came up with a lot of colors. When you have a lot of colors, there's lots and lots of wonderful things to do. And Jeff Lichtman and Josh Saines developed this wonderful way of using these colors to look at different nerve cells in the brain and to start to look at the connectivity of the mouse brain, which they named this technique Brainbow. But I want to get back to this question of how do we make discoveries? Are they just always accidents while we're doing other experiments? Well, obviously we have to do lots of experiments. But I think that part of it is to always be questioning, to always be wondering about the world. And in particular, I think we should be questioning things that are actually right in front of us all the time. And I've made a little collection of some of these ideas or things that are always there, but I very rarely found anyone that ever questions them or wonders about them. And yet the questions are all around us. And I think we should also be questioning our assumptions. So what do I mean by things that are all around us? Well, my first scientific paper involved work that involved the major part of the body that's transparent. And when I give talks, and when there's an audience that I can see, I often ask people to raise their hands if they know what part of the body is transparent. And I can tell you that only about 5% of the audience raises their hands. Most of the people look and they have no idea where the transparent part of the body is. And then I give them a hint. And I say, it is right in front of your eyes, because it is the cornea. Even though maybe about 5% of the people that I've in my audiences will raise their hand and will realize that there are transparent parts of the body, the cornea, the lens. I have never met anyone who asked the question, or I said, you know, I've wondered about this for years. Why is it that this tissue is transparent, that we can actually see through it? And that's a fascinating question. I had not thought of it when I went to work in the lab that I did, but fortunately someone had. Another one of these questions, things that we see every day, but we sometimes don't ask questions about, has to do with plants and their growth with regard to gravity. We know that when a plant grows, the root goes down into the ground, the shoot goes up into the sky. We know from experiments that were old when Darwin repeated them, that if you take a potted plant and put it on its side, the roots will turn to grow down and the shoot will turn to grow up. But I don't think people have realized that this is not the only response to gravity of the plant. And in fact, it's not just the roots and the shoot, but in fact, all the parts of the plant. That branches, for example, in a tree, have what's called a set point. That is a particular angle at which they grow in relationship to gravity. And they have this same thing. So we have some trees, like the cypress, in which the branches all go straight up. Others where they go up at an angle, others where they go off parallel to the ground. And some of them are mutant and don't grow up at all, but simply droop because they can't respond. This is sort of going to ruin your life because you're never going to be able to look at trees again without realizing this idea of the set point and how these are all different. And there are people that are working on this problem. Let me give you the third problem. The third example comes from when I was a graduate student, I helped teach a course in physiology. And the instructor would be talking at one point, would be talking about muscle. And he would say, you know, there are two types of skeletal muscle. One type is called the fast twitch. It responds quickly, but it can't keep going. It can't be sustained. And it goes into oxygen debt. And then he said the other type of muscle is one called a slow twitch. This can work over a long period of time. Doesn't go into oxygen debt because it brings its own oxygen in. It does so with a molecule like hemoglobin, called myoglobin. Now, I can tell you that most of the students in the class by this time had fallen asleep listening to this information about muscle. And then he said, so the next time you have chicken or turkey, now you can understand the difference between the white meat and the dark meat. Because in fact, one is fast twitch muscle and the other is slow twitch. Again, I've ruined your life because you're going to be thinking about this every time you have chicken or turkey again. But these are all things that are right in front of us that I certainly had not ever asked these questions. There's lots of different things in our lives that could be the basis of very interesting studies. I want to get to this question of questioning assumptions. When I was younger, actually, I still am sort of excited about these matchstick puzzles, which were puzzles that said, okay, you have this pattern of matches. Could you move 3 matches in this set to make 3 perfect squares? And after some time, you might be able to say, okay, let's move these 3, and now we can get these 3 perfect squares. Another one of these puzzles is move just 3 matches and turn the fish around. And after some playing around, you maybe get to this position here where the fish now is going in the opposite direction. But then you get a puzzle like the following. Here are 6 matches. Use the 6 to make 4 triangles, 4 equilateral triangles. And as much as you rearrange them on the paper, it may be very difficult to do that until you realize that you have assumed that you have to work in 2 dimensions. And if you get rid of that assumption and you use 3 dimensions, then you can make 4 equilateral triangles with the 6 matches. There's actually another assumption. The assumption, why should the triangles all be the same size? If they're not, then you can lay them here so that you can actually get 4 equilateral triangles. 2 are of one size, 2 are of another. And to even carry this further, there can be the problem of make these 3 matches into 6. Well, yes, you could break them, but without breaking them, can you change these 3 matches into 6? And the assumption there is that we're thinking about shapes. What if it's not shapes? What if it's Roman numerals? We can just move one over and now we can get 6 made by the 3 matches. So all of these are asking us to really question what we've been assuming in our work. Now, I have a good friend, Muming Pu, who's at Berkeley and also head of the Neuroscience Institute in China. And Muming's career has been founded on questioning what we have previously assumed. He calls it the retrospective approach. Now, what he does, and he's done it spectacularly, he reads an elementary textbook. Now, for him, the textbook was the 2nd edition of Molecular Biology of the Cell. And he reads what's written. This is what people believe. And then he says, okay, why do they believe that? And he usually finds out that it's based on some very old experiments. And we have now much more modern methods and different ways of thinking about it, and much more information. And so he retests the hypothesis using these modern methods. And he usually finds that it was wrong. So he publishes a wonderful paper that corrects this. And he's done this his entire scientific career and has done a great job. So it's not that you get at the most arcane problems, but what are the real foundations of what we believe? Now, I want to take questions, but I also want to end with my favorite scientific quote about basic research. This quote was by a physicist, Robert R. Wilson, who in 1969, now this is when the United States was fighting a war in Vietnam. There was a lot of concern about defense, about security, and Robert Wilson was asked to talk to Congress because he was in charge of a group that wanted Congress to pay money to build what became the Fermi particle accelerator, the largest particle accelerator in the world until the CERN super collider came about a few years ago. But in 1969, Congress was trying to debate whether it was important to do this. And so he was asked to come to a hearing. And at the hearing, a senator who was very much in favor of this thought he would ask him an easy question. He said, Dr. Wilson, can you tell us all the ways that this particle accelerator is going to help national security? And Wilson gave him an answer he didn't want to hear, none. So he asked the question again and again, and he always said none. And finally, the senator said, Dr. Wilson, really tell us, in what respect will this particle accelerator help the national defense? At which point, Robert Wilson said the following: it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
Thank you, Professor. So we can start from we have one question from Yuri. Dear Professor, do you believe that protein cells and generally biological systems can be programmed like computer?
So people have been able to do various logic circuits within cells, change with DNA. Several years ago, we never published on this. We thought about different ways that one could use GFP to make logic circuits with these things. I'm not really that knowledgeable of this area, so I can't really tell you what the state of the art is, but this is something that people are thinking about a lot.
Well, okay, thank you. Then the next question, for our young scientists, what do you recommend how to, to be the scientist is very hard and a very long road away to become a great scientist. What do you recommend for young students to become, and do not lose motivation for this?
So I think, so there's a number of answers to that question. I think that one of the things that people are sometimes most frustrated about is that their experiments don't work. And so they get discouraged. And it's very hard not to be discouraged when things don't work. I have a particular approach to that. And it comes about, and part of what I want to tell you has to do with something that happened quite a long time ago, 2004. I was being inducted into the National Academy of Sciences here in the United States. And at the ceremony, my wife and daughter, my daughter was 12 years old at the time, were in the audience. Now, the induction ceremony is an exceptionally boring ceremony. And the reason it's boring is because there are 70, now there's over 100 people every year that one at a time are introduced to the audience. But the people in the audience only care about one of those people, but they have to sit through everybody. And the one person gets up on the stage, shakes hands with the president of the academy, has a picture taken with the president of the academy, walks across the stage. There's a book that people sign. And while they're walking across the stage, they are, somebody reads one or two sentences saying what that person had done. Well, I went and did that. I went through, eventually I got back to my wife and daughter, and I knew my daughter, being 12, was probably not very excited about being at the ceremony. But I told her that I thanked her, and I said it was really nice having her there as support. And I said, but you must have been very, very bored. And she looked at me, as she often looks at me, like I am the stupidest person in the world. And she said, Dad, don't you understand about those people? So I said, okay, what about those people? She had been listening to all those sentences. She said, Dad, each one of those people was the first person in the world to see or do what they did. The first person. Now, I'm exceptionally proud of that because that's what we do as scientists. We're the first ones in the world to see something, to add that element of knowledge that no one else has been able to get before. And because we're always working at the unknown, that's really hard. And it's frustrating, and we don't always get it right all the time. If it was easy, people would have done it already, but it's not. So it's frustrating because of what we're trying to do, and that is make discoveries. So I think for the part of it is the realization that things are not going to work all the time. It does mean that you have to put a lot of effort into it. And I think it is sometimes useful to be working on more than one thing. Often people that have labs are a little happier than the people in their labs because each individual person might be working on one project and they may or may not be successful. But if you have several people in the lab, there's at least one person who's succeeding and you can get excited about that. Well, you can do that individually too. You can work on more than one thing. Although nobody likes to hear someone say, do twice as much. But I think having an alternative that you're working on is sometimes a very good approach to overcoming the frustrations that are part of scientific endeavor.
Thank you. The next one, what do you think about the dynamics of the science development in Ukraine? How the situation has changed over the 30 years of independence? In the point
So, I'm unfortunately too ignorant of science in many countries, and I don't know the situation in Ukraine. The only I have a very old view of a scientist from Ukraine because I'm here at Columbia University, and Theodoros Dobzhansky was one of our most famous faculty members, a Ukrainian scientist who was really the founder of population genetics here at Columbia. I unfortunately am too ignorant of the state of the science. That's something I need to learn more about.
Okay, thank you. Do you think useless knowledge promotes science? And do you think science needs to be promoted?
I think that there is, I think people appreciate science. I think in this time of the pandemic. People have appreciated it a lot. I think that we see it in our daily lives, but I think people don't recognize all the background, so-called useless knowledge that needs to be acquired for us to make strides ahead. Understandably, if you have a loved one who is suffering from a disease, it's not easy to hear. We need to have more general knowledge so that we can get new approaches to all diseases, and in particular, this disease that you're very concerned about, because your loved one is afflicted and you want to have something that, so I think there's a natural inclination among non-scientists, for example, with regard to health, of don't we have, the cure is the thing. But in fact, the cure is part of a long line of scientific research. The same thing happens with global warming. People want to see solutions. Too many of our movies sort of have scientists, oh, we have a problem, here's what we do, here's the answer to the problem. But in fact, the work really requires having background information. No one came to Charles Towns. Charles Towns got the Nobel Prize for work he did here at Columbia, where he developed the maser, which led to the laser. And no one came to Charles Towns when he was doing his research and said, Charles, I want an instrument that will allow me to do surgery in a very fine way, maybe on the eye. Or I need to have a way of putting music or videos on a disc and being able to read them. Or I want to be able to get people to check out of the grocery store more rapidly. But all of these applications from the laser came about after it was invented. There's also been an enormous amount of basic science that came about because of the laser. Low temperature physics, gravity waves, the laser tweezers, all of the won Nobel Prizes that are impossible without the basic research from the laser, and wouldn't have even been imagined. But once it was there, it allowed for other things to take place. We see this all the time in biology, where people are thinking about things along the path that they already know about, and then someone makes a new discovery, whether it's CRISPR-Cas9 or GFP or microRNAs or interfering RNAs. All of a sudden, there's a new perspective, a new way of thinking about the old problems. The problems stay around, but we need the new approaches to address them.
Okay, thank you. That was the last question. Thank you very much for your time. I'll make a little announcement in Ukrainian. Дякуємо всім учасникам за лекцію. Сьогодні для вас доповідав Мартін Чалфі, і для реєстрації ви можете зайти на оновлений сайт mind.gov.ua і побачити наступний розклад на наступні лекції. Thank you very much, Mr. Chalfie.
Thank you. Take care.